Hi everyone, I'm Sreedevi. I'm doing my PhD in Department of Neurochemistry at NIMHANS under the guidance of Dr. Gokulakrishnan. My topic for today's presentation is exploring metabolomic biomarker signatures in externalizing disorders, insights into neurodevelopment and cognition. First, talking about what are externalized disorders. So there are different ways in which the emotional and behavioral problems can manifest in children or adult. Like some will be thought related symptoms, some will be detachment symptoms. However, the very common ways in which these behavioral problems are manifested are externalized and internalized symptoms. However, as the name suggests, in internalized symptoms, the symptoms are mostly inwardly directed, like the examples will be fear, anxiety, everything. But coming to externalized symptoms, they can be defined as problematic or disruptive behaviors related to aggression, impulsivity, inattention, and hyperactivity. So the examples for this externalized disorders are attention deficit hyperactivity disorder, oppositional defiant disorder, conduct disorder, etc. So all these are externalized disorders or externalized symptoms that are manifested in the childhood. So if they are manifested in the childhood, there is a chance that it can lead to other externalized problems in your adulthood. And the examples for them are substance use disorders, impulse control disorder, adult ADHD, violence related symptoms, or they will also have poor performance in their academic and professional field, etc. And studies have reported that this externalized disorder have a prevalence ranging from 7 to 10%. So considering this high prevalence rate and the problems that they can cause in the person who are affected with externalized disorder, that has to be considered as a serious issue which has to be addressed. So how are we going to address this? With the help of biomarkers. So there are different types of biomarkers from your genomic study, transcriptomic study, everything. And here we will be concentrating mostly on metabolomic biomarker. So this, why metabolomic biomarker? Because it is more closely related to your disordered state, and it is mostly affected by other factors like your diet, the way you are giving birth, the gut brain, the gut microbiota, everything. So here we will be targeting on the plasma biomarkers because of major two reasons. The first thing, it is non-invasive, and the second one, it is easy to monitor throughout the span of your disorder or condition. So, and coming to psychiatric disorder, many studies have shown the relevance of plasma biomarkers. So I have two examples with me in which a study has shown a panel of metabolites like L-cysteine, of phosphoethanolamine in patients with ADHD. And also, I have another study in which they have shown that aspartate, creatinine, etc., is shown to vary in autism spectrum disorder. So here we hypothesize that there will be some metabolite signature in patients with externalized disorder, which we can use to study the process of neurodevelopmental pathways and cognition. So what is the lacunae in the current field, and what is the rationale of my study? So currently there are no reliable and robust panel of biomarker or a single biomarker which is associated with externalized disorder in the Indian population. Even though that is available in other countries or other population, that cannot be extrapolated to our Indian scenario, because there will be, of course, variation in the genetic, environmental, and socio-cultural factors. And also, there are no studies as of now that correlate the metabolomic signature that we get along with the brain cognitive and the imaging data. So considering this research gap, we are planning to conduct a biomarker study leveraging samples from c-VEDA cohort. So c-VEDA is nothing but consortium on vulnerability to externalized disorders and addiction. So the c-VEDA is a cohort that was established in India with the help of ICMR and UK Medical Trust in the year 2015. So the major aim when they started this c-VEDA cohort is that they wanted to assess they wanted to simultaneously assess behavior, brain function, brain structure, other biological and environmental factors, because all these factors together are contributing to externalized disorder. And as I mentioned before, even though this similar kind of studies have been done in high income countries, mostly in Australia, UK, everything, this data cannot be extrapolated to India because of the variation in genetic environmental factors. Considering all these aspects, c-VEDA is India's first and largest longitudinal study offering an in-depth characterization of psychopathology, cognition, brain structure, function, environmental factors, etc. So current study is a part of c-VEDA project. So the objectives of my study are to identify a metabolomic signature or a panel of biomarker which can correlate with neurodevelopmental and cognitive environment by using absolute IDQP 400 high resolution kit in the plasma samples from c-VEDA cohort, which have undergone a deep phenotyping data, and to also perform a fatty acid profiling among the participants using GCMS in the plasma samples. And the final objective is to correlate this metabolomic biomarker with the existing neurocognitive and neuroimaging data to understand the susceptibility towards neurodevelopment and cognition. So this is the c-VEDA cohort. So talking about c-VEDA cohort, they have collected collected the samples from 9010 participants from different sites in India. So all these different sites are under different socio-economic conditions. Like suppose in one sample site, they have parents who have undergone parents who already have some psychiatric conditions. In other region, the people who have undergone the study have undergone some socio-economic problems, etc. So in this, around 1883 samples were collected at the NIMHANS, and of that 1883, 538 people have undergone deep phenotyping assessments, which include cognitive assessments, MRI scanning, and other genomic scanning. So I'll be targeting on this 538 people who have undergone this deep phenotyping, on which I'll be doing my metabolomic analysis. So I have done the sample size calculation, for which so for small effect size, the sample size is around 496, and I consider the c-VEDA cohort is ideal for the study. So this is my study design. So I have the c-VEDA biorepository of plasma samples, in which I have 538 samples. So with this 538 samples, I will be doing the processing, one for getting the polar and non-polar metabolites using the absolute IDQP 400 kit, and the other one for fatty acid. Yeah. So I'll be trying to find out a single or panel of biomarkers with which I'll be correlating it with the cognitive and MRI data that is already available with the c-VEDA. So right now, as a preliminary study, I have done the plasma sample extraction for both polar and non-polar metabolites using methanol and MTB extraction respectively. And I have done I have used helic column for polar analysis, and I have used a reverse phase column for non-polar analysis. And these are the preliminary results from my lab. So I have standardized a method for just standards right now for both non-polar and polar analysis. So in non-polar analysis, I have standardized the method for sphingomyelin, ceramide, cholic acid, and indole propionic acid. And coming to polar, I have standardized the method for tryptophan and kynurenolic acid. And I have coming to polar metabolites, I had targeted total 150 compounds, and I was able to obtain total 125 compounds out of that. And coming to polar, I had targeted around 175 compounds, and I was able to obtain these many compounds in my analysis. So right now, from these preliminary data, I'll be going for targeted analysis using the IDQ kit for both polar and non-polar compounds. So this is the statistical analysis I'll be doing. I'll be converting the continuous variables, and I'll be comparing the study groups using appropriate statistical tests, and we'll be running the bootstrap enhanced penalized regression for the variable selection, after which I'll be doing ROC analysis to understand the sensitivity and specificity of the selected biomarker, and a level of significance of less than 0.001 will be considered as statistically significant. So what is the significance of this study? So this will provide a first-line clinical data to understand the extent to which how a metabolomic signature can influence the neurodevelopment and cognition. And it can also, in other way, help us to understand the pathophysiology of externalized disorder and also the associated molecular pathway. And this selective panel of biomarker in a long run can also help us to understand, carry out early diagnosis, early treatment, everything. And this is the overall summary of my work. So right now, I am searching for a novel biomarker, because there is a lack of biomarker which is in externalized disorder, which can correlate with neurodevelopment and cognition. And there is lack of translational research in the current field. So I'll be coming up with a novel biomarker with which I can correlate it with brain structure, physiological states, brain function, and cognitive performance. So I thank NIMHANS, ICMR, and c-VEDA for helping me in this research. Thank you.